Hello, this short video will provide you with a method for performing Agarose gel electrophoresis, a common molecular biology technique used in research laboratories. The genome of a cell, made up of DNA, contains all the heritable information about how cells are run. Each organism has a unique genome. This is why DNA comparison can be applied to identify familial relationships, criminals, and diseases. The size of fragments of DNA can be identified by a separation in a gel matrix. The gel has small pores through which the DNA can travel. DNA is negatively charged due to its phosphate backbone. Thus, it can be driven through the gel by an electric current. The DNA will pass through the gel matrix from the negative electrode to a positive electrode. Remember that opposite charges attract and like charges repel. The larger the fragment, the slower it travels through the gel due to resistance. First, agarose beads are weighed and combined with enough of the running buffer, in this case TAE buffer, to make a 0.8 concentration of agarose. Concentrations may range from 0.5% to 1.2%, depending on the size of the fragments you wish to resolve. Next, the mixture is boiled in a microwave to melt the agarose bead. Once the gel has cooled to about 30 degrees Celsius, it can be poured into the mold, which contains a comb that will form the wells when the gel solidifies. While the gel cools and solidifies, the DNA samples can be prepared. Normally, a loading dye is added to each sample. Because we are using a loading dye that also stains the DNA such that it will fluoresce under UV light, we must also add this dye to the latter sample. If we were staining the whole gel with ethidium bromide, a mutagenic chemical that stains all DNA in the gel, we would not have to worry about adding loading dye to the latter. The latter is a standardized solution of DNA containing known DNA fragment sizes to compare to. Once the gel is solidified, the mold tray is placed into the gel apparatus containing running buffer. Buffer is added to cover the gel surface, and the comb is removed. First the latter, then the prepared samples, are pipetted carefully into the wells of the gel. After all the samples are loaded, the cover of the apparatus is placed such that the electrodes match up, always red to red and black to black, and the cords are plugged into the power supply. The voltage and timer are set on the power supply, and it is turned on. Tiny bubbles should form in the buffer if the apparatus is set up correctly. Remember that DNA travels from negative, black, to positive, red, and so the wells should be on the negative end of the apparatus. As the gel runs, the dye will move through the gel, creating a dye front. Dyes are sometimes selected on their molecular size so that a size of DNA can be tracked. In general, you don't want the dye front to run at the same speed as your fragment, or else it will cover up the band you are looking for. Depending on the size of the bands you are expecting, you may stop the gel when the dye front gets to a certain point along the gel. The non-toxic dye we added to the samples mimics the effect of ethidium bromide and fluoresces under UV. A photo must be captured using UV light in a computer program. The brightness, contrast, and exposure are adjusted on computer programs, and this is the final image of the gel. On the left, you can see the DNA ladder with known molecular sizes. In the lanes on the right, you can see the fragment sizes contained within each sample. This has been a video on how to resolve DNA fragments in an agarose gel. Thanks for watching.